Around 800,000 years ago, a group of early humans left their African homeland on a journey that would change the course of human history. Over time, this group evolved into three distinct types of humans: our own species, Homo sapiens, Neanderthals, and Denisovans. Just like a family splitting up, some relatives moved west of the Himalayan mountains and became Neanderthals. Others went east and south to become Denisovans, while our direct ancestors stayed in Africa. Over the past 20 years, scientists have made incredible progress in studying human DNA. They've collected vast amounts of genetic information by analyzing DNA from both ancient human remains and modern humans. The challenge has been making sense of all this data. Imagine trying to piece together millions of family trees from different sources all at once. Now, scientists from David Reich Labs at Harvard University have developed a clever new way to handle this massive puzzle. Their method creates a giant family tree that can handle millions of DNA sequences at once. This tree has 231 million branches, each representing different family lines throughout human history. Incredibly, this is the largest genetic family tree ever created, providing an unparalleled view into our ancestry. To build this tree, they started with 8 complete ancient human DNA samples as the foundation. They then added thousands of smaller DNA pieces to fill in the gaps and confirm the connections. They also included DNA from 3 Neanderthals and 1 Denisovan, our ancient relatives, which showed slightly older genetic patterns than modern humans. Each section of the human genome comes from just one parent, letting us trace specific genetic variations back through time like branches on a family tree. This new research shows how all humans worldwide share these genetic connections. As Dr. Anthony Wilder West from MIT and Harvard explains, we're reconstructing ancestral genomes to create a vast network of relationships, helping us pinpoint when and where our ancestors lived. Researchers studied DNA differences that make each human population unique. They found 6,412,077 genetic variations and traced when and where each one first appeared. While some ancient DNA samples couldn't be used in the main study, these 3,589 samples still helped scientists understand when different genetic traits emerged throughout human history. Recent research has uncovered ancient DNA patterns over 72,000 years old, primarily found in Northeast Africa, with the oldest 100 genetic variations traced to modern-day Sudan. These genetic markers date back nearly 2 million years, far older than Homo sapiens, which emerged around 300,000 years ago. Interestingly, these ancient variations connect to Homo erectus, suggesting that early human ancestors first evolved in the Sudan region before later migrations, and intermixing shaped modern human DNA. The discovery of these markers challenges the traditional view that Homo sapiens solely evolved in a single region, suggesting a more complex, multi-regional origin. A study conducted in 2019 provided deeper insights into African genetic diversity, particularly among the Khoisan people, who carry one of the most ancient mitochondrial DNA lineages. To trace its origins, researchers analyzed the mitochondrial DNA of 1,217 individuals from Namibia and South Africa, grouping them by ethnicity and language. Their findings revealed that early human populations remained stable for about 70,000 years, likely supported by Botswana's ancient wetlands. However, between 130,000 and 110,000 years ago, genetic diversity surged, suggesting that newly formed green corridors encouraged migration and expansion. This period of increased genetic diversity may have coincided with the development of early symbolic behavior, including art and jewelry, which are among the earliest known examples of human creativity. To further map human ancestry, scientists analyzed modern and ancient human genomes from 8 databases, compiling 3,691 genome sequences from 215 populations. Using advanced computer algorithms, they reconstructed evolutionary trees, tracing common ancestors, and identifying patterns of genetic variation. This extensive analysis uncovered nearly 27 million ancestral connections. After incorporating location data, researchers were able to estimate where these predicted common ancestors once lived. According to the evolutionary tree, Early humans first appeared in what is now Sudan around 2 million years ago. They expanded into the Middle East about 840,000 years ago and reached southern Eurasia by 280,000 years ago. By 140,000 years ago, human populations had spread across most of Eurasia and into Papua New Guinea. The ancestors of Native Americans had already branched off as a distinct lineage by 56,000 years ago, and modern humans had settled throughout the Old World. Researchers have found that the Papua region today retains the highest proportion of Denisovan DNA, along with significant traces of Neanderthal ancestry. These findings provide insight into the complex genetic history of early human populations. In his 1859 book On the Origin of Species, Charles Darwin emphasized natural selection as the key driver of evolution. Other evolutionary processes, such as genetic drift, played a role as populations separated over time. The founder effect influenced genetic diversity as small pioneering groups established new settlements, while cultural and sexual selection further shaped human variation. In The Descent of Man, Darwin keenly observed the role of sexual selection in human evolution. However, reconstructing the appearance of ancient human ancestors remains challenging, with ongoing debates about their exact features. Early humans who lived in diverse environments a million years ago were likely as varied in skin color and hair type as modern humans, perhaps even more so. Some ancient populations may have had dark skin with blue eyes and blonde hair, much like present-day Melanesians. Since many early human groups have gone extinct, it's a mistake to assume that traits like blue eyes or blonde hair were exclusive to Europeans. French anthropologist Ludovic Slimak has criticized modern reconstructions of ancient humans, arguing that we often impose our own biases, turning them into Frankenstein figures shaped by contemporary assumptions. Some research suggests that certain genetic traits, such as resistance to altitude sickness, were inherited from archaic humans. Tibetans, for example, carry a Denisovan gene that helps them survive at high altitudes with lower oxygen levels. Despite these challenges, scientific estimates of ancient human appearances have remained largely consistent. Neanderthals in Central Asia and Europe likely had lighter skin pigmentation than those living closer to the equator. Genetic studies show that Neanderthals exhibited a range of pigmentation traits for skin, hair, and eyes, some of which still exist in modern populations, with lighter pigmentation linked to certain inherited Neanderthal genes. A study from the University of Pennsylvania found that skin color variation existed long before Homo sapiens. Researchers identified 7 genetic variants linked to lighter skin, with some emerging at least 270,000 years ago, and 4 dating back over 900,000 years. This means the genes for lighter skin were already present in early hominin ancestors, long before modern humans appeared around 300,000 years ago. The presence of light skin alleles in early modern humans, Neanderthals, and Denisovans suggests they inherited this trait from a common ancestor. For at least 1 million years, both light and dark-skinned human populations have coexisted. The study also pinpointed 8 genetic sites responsible for about 30% of skin pigmentation variation, each containing variants for both lighter and darker skin tones. 7 of the lighter variants emerged at least 270,000 years ago, while 4 date back over 900,000 years. Some of these variations appeared around 500,000 years ago, suggesting that early modern humans and Neanderthals may have had skin tones similar to Eastern African pygmy or sand populations. The darkest skin tones typically result from 2 mutations that reduce gene expression, indicating that darker pigmentation evolved as a derived trait from lighter skin. This is consistent with observations of light-skinned chimpanzees beneath their fur. Most of these genetic variants have ancient origins, likely first appearing in Homo erectus. These genes coexisted across different latitudes for over 1 million years before human species diverged, leading to a range of skin tones. Light and dark alleles have been present in the hominin lineage for hundreds of thousands of years, with ancestral variants also found in Neanderthal and Denisovan genomes. Researchers discovered ancient genetic variants linked to light skin, eyes, and hair in the Sand people, variants found in 2 interconnected genes, HERK2 and OCA2. Though commonly associated with European traits, these genes actually originated in Africa around a million years ago before spreading to Europe and Asia. The blue eye mutation likely emerged in the Caucasus region about 42,000 years ago, caused by a change in HER2 that suppresses OCA2 expression. This trait gradually spread westward into Europe around 20,000 years ago and became widespread by 10,000 years ago. While up to 16 genes influence eye color, OCA2 and HERC2 on chromosome 15 play the most significant roles. Today, we know over 100 genetic variants linked to pigmentation in modern humans, while fewer than 10 have been found in Neanderthals. Two Neanderthal haplotypes associated with pigmentation are now widespread in European populations, suggesting they were favored by natural selection. One of these haplotypes includes the OCA2 gene, which plays a key role in determining eye color, particularly the distinction between blue and brown eyes. This Neanderthal-derived OCA2 haplotype highlights the complexity of tracing genetic traits passed down through ancient interbreeding. Notably, this haplotype contains no coding variants, meaning its effects likely stem from gene regulation rather than changes in the gene structure. Among the many associated variants, only RRs140879 has been previously linked to pigmentation. While this variant has a small but measurable impact on eye and hair color in people of European ancestry, its weak predictive power for eye or skin pigmentation suggests that other, yet undiscovered linked variants may be responsible. These findings provide strong evidence of recurring positive selection acting on multiple OCA2 variants, some evolving independently in modern humans, while others were introduced through interbreeding with Neanderthals. And with that intriguing discovery, we leave you to reflect on the fascinating complexities of our shared human ancestry. Until next time, stay curious and keep questioning. If you enjoyed this, don't forget to like, share, and subscribe for more insights into human evolution and genetics.